He kōna I purangi tēnei nā te reo irirangi o Aotearoa.
It's a horrible disease. A large number of people are diagnosed when the treatment options are limited, when it's too late for surgery. And, we do see younger people being diagnosed with cancer in their 40s, 50s, when they're still primary income earners for their family and they've still got a young family. And so, it's really, really tragic. And so, if we can just reduce the rates of stomach cancer a little bit, then, that's making a huge leap in healthcare.
Kia ora, nau mai haramai ki te ao hurahanga. Hello and welcome to Our Changing World. Ko Concannon tēnei. Dr. Tom Mules kind of wears two hats. And in one, he's come across several patients with stomach cancer.
I'm a gastroenterologist at Hutt Hospital. So my role out there is to look after people and treat people with gastroenterology problems and liver problems, and also perform endoscopies. And then here at the Mulligan Institute, we're establishing a gut health program looking at a number of issues, one of which is Helicobacter pylori. But really what we're trying to do is translate many of the discoveries that we make here into the clinic so we can make big changes for our patients.
Helicobacter pylori is a bacterium that infects your stomach. It's very common. 50% of the world's population are infected with it, but most people never realise because they don't get symptoms. But it can damage the tissue of the stomach and cause inflammation, and in some cases peptic ulcers. It's also implicated in stomach cancer.
Stomach cancer is a relatively common cancer. Worldwide, it's very common, particularly in eastern countries. In New Zealand, it's less common. It's the 8th or 9th most common cancer, or cancer killer. But actually looking at those numbers isn't that representative. If you actually look more closely at different ethnicities within New Zealand, so MÄori and Pacific, it's the 4th leading cancer killer in New Zealand. And so it's a huge, huge problem, and it's a huge problem that's causing inequity within our healthcare system. Those populations have a 3 to 6 times higher rate of stomach cancer compared to New Zealand Europeans in New Zealand. And one of the key reasons, or the biggest reason for that, and the number one most modifiable risk factor for stomach cancer, is Helicobacter pylori.
Why it can cause stomach cancer in some people but not in others, we don't know yet.
Being able to work out who is going to develop stomach cancer is very, very difficult. But we know there are some risk factors which may come down to genetics, other environmental risk factors which increase the inflammation related to Helicobacter pylori. But we also know there are different strains of Helicobacter pylori. And Helicobacter pylori in Asian countries is a much bigger problem, and they have much higher rates of stomach cancer because they have strains of Helicobacter pylori that are much more carcinogenic, so lead to the development of cancer. And that's one of the reasons why we have such different rates between ethnicities in New Zealand, because MÄori and Pacific are not only more likely to be infected with Helicobacter pylori, but are more likely to be infected with the carcinogenic strains of Helicobacter pylori. So they kind of get a double whammy there.
50% of the world is infected, but it's not the same across all countries. So what does the rate look like in New Zealand? Well, the best data we have comes from a small South Auckland study from a decade ago. Looking at just shy of 600 people, the researchers found that around 30% of MÄori and Pacifica had Helicobacter pylori, while for New Zealand Europeans, it was just under 8%. Something that Tom wants to do is update this data, so we have a clearer picture of the scale of the problem. But why the difference in infection rates?
Helicobacter pylori is acquired during childhood, and typically it's thought to be from your parents or the people that you live with. And so that comes back to your socioeconomic conditions, so like lots of infectious disease, living close together, sharing beds, sharing food, are all risk factors for Helicobacter pylori. But actually, probably the biggest risk factor is if your parents are infected. So that means it does get passed down through generations, and the same strains get passed down through generations as well.
So that more carcinogenic strain is likely just to get passed down through generations.
That's right. And it's really interesting. You can look at migration patterns and look at the strains of Helicobacter pylori amongst different ethnicities within New Zealand and can trace it back to where they've migrated from when they first came to New Zealand.
So we've got a bacterium implicated in cancer that's contributing to health inequities in Aotearoa. But it's a bacterium. We've got antibiotics for that, right? And the answer is yes, but...
To treat Helicobacter pylori, we give patients a course of antibiotics, and that includes 3 different pills that you take a number of times a day for a 2-week period. And currently in New Zealand, we are giving everyone the same treatment once we diagnose Helicobacter pylori. We call that triple therapy. However, internationally, we know that the rates of antibiotic resistance for Helicobacter pylori are increasing. What we don't know in New Zealand, and it comes back to what I said earlier, that our last data around antibiotic resistance was about 10 years ago. 10 years ago, the rate of antibiotic resistant to the antibiotic clarithromycin, which is the main one we're interested in, had doubled since 1999. So it was already on the dramatic increase. What it is now, we're not really sure, but perhaps it has doubled again. And so that means that we're seeing more treatment failures within our clinic. And so what we're trying to do is to streamline that process and give the right antibiotic for the right person so we can cure the Helicobacter pylori.
So in the clinic, you're using this triple attack treatment.
Yeah.
You're sending people off with these antibiotics, but they're coming back and the bacteria is persisting. And you're like, okay, something is awry here, and maybe it's that increase in antibiotic resistance.
That's right. We still cure a lot of people with the triple therapy, but the number of people we're curing is probably reducing, and taking 2 weeks of antibiotics and then getting told that your antibiotics hasn't worked, that's pretty devastating, because then the next step is prescribing even more high-powered quadruple therapy, so 4 different pills for 2 weeks. And so being able to prescribe the right antibiotic course first up is a much more attractive option for a person, and is very important in terms of not prescribing antibiotics incorrectly. So it comes back to this idea of antibiotic stewardship. So prescribing the right antibiotic for the right person the first time so we can treat their bacteria.
To do this, you first have to figure out how to easily and accurately test for whether the bacteria has resistance or not. Downstairs in the labs, Kate is working on this.
Hi, my name's Kate McLean. I'm a clinical researcher and clinical trial coordinator here for the H. pylori project at the Mulligan Institute.
And today you are extracting DNA from some tissue samples.
Yes, so we've got some samples that were taken from patients who had to get a gastroscopy procedure, so they had to get a camera down into their stomach, and some little stomach tissue samples were taken. And so we've got those. We're going to get the DNA out and see if we can detect the DNA of the bacteria, the H. pylori.
Because you're not interested, you're going to extract DNA, but you're not interested in the patient's DNA. You're really focused on whether that bacteria DNA is there.
Yeah, so we won't really be looking at the human DNA at all. We'll do a little test of just a generic human DNA gene that just tells us if it's human, to make sure we've gotten DNA out of the sample. But after that, we'll just be looking at genes that are specific to the bacteria to see if it's in there.
And when you zone in on that bacterial genes, you're also going to do a test to see whether it's got antibiotic resistance, right?
Yes, yeah. So there can be some mutations in the DNA of the bacteria. That means that it won't be or isn't as likely to be killed by antibiotics. So we want to see if we can detect those tiny little changes in the DNA that would then tell us whether someone's treatment with antibiotics is likely to work or to not if it's got that mutation.
And how do you go about doing that?
So we use a method called PCR, that stands for polymerase chain reaction, which doesn't really mean a whole lot, but essentially we amplify up the DNA, expand it so it's easier to detect, and then we use a probe that attaches to a specific sequence of DNA that we are interested in that codes for that mutation, and we can detect that through our machines using fluorescent light. And so the amount of that fluorescent light that we see would determine whether it has that different sequence that we're interested in or not.
So you hone in on the piece of DNA that potentially has that mutation for antibiotic resistance. You make lots and lots of copies of that so that you've got heaps of copies, and then you chuck in the probe, which will attach only if that mutation is there, and then run it through a machine and see if it lights up.
Exactly, yeah.
Okay, cool. That sounds really cool.
It's very exciting, yeah. It's cool. The way DNA works is kind of like a zip, so it clicks into the matching sequence on the other side, and that's what's attached to the light, yeah.
And I mean, all of this, like I say, it's really cool, but you can't really see any of this happen. This is all happening on a tiny, tiny level.
Very much, yeah. It just looks like liquid in a tube to me at all. You can't see the DNA itself at all, but it's cool when you can see the graphs on the computer at the other end. You can see the DNA increasing in the form of a line, I guess. So yeah.
And at this stage, are you kind of just working through the methods to see whether it all, works to be able to identify this antibiotic resistance?
Essentially, yeah, because at the moment this test isn't being done in New Zealand for this particular bacteria. Normally, to check for antibiotic resistance, they'll grow up the bacteria on a little agar plate in a petri dish, but this bacteria doesn't like being grown, so it's just not done. So we're trying to see if this DNA-based method is something that could be feasible to do in hospitals, in hospital labs in New Zealand, yeah.
But it is being used overseas, so this method has been developed and used and tested in other places, and you're just kind of checking to see whether you can make it work here in this lab.
Yeah, it's still pretty early days with it being done overseas. It's not standard practice everywhere, it's still pretty in early development, but it's beginning to, so we want to see if we can bring that over here to New Zealand, yeah.
And these samples that you're working with today, I mean, when it comes to you, it's kind of just a little bit of tissue, but they've each come from a person, right?
Yes, exactly. And that's the really important thing to remember, is that it just looks like a squidgy little thing in a tube, but it's actually from a real person who's been sick and needs to find out what's going on. And hopefully with developing these methods, it will make the testing a lot easier and help a lot more people in those situations.
She's in the middle of her research as I chat to her. We collect the samples from another lab.
So I've got some little bits of stomach tissue in each of these tubes, and they've just been shaking away in the warm for the last couple of hours to dissolve away the tissue and let the DNA out. Okay, cool. So I'll take them back through to the other lab.
Then they get popped into a centrifuge, which spins the tubes hard. So the centrifuge is basically spinning the tubes really fast, and that's going to separate out, like, the heavier things are going to settle to the bottom, and then the liquid will be at the top. So where will the DNA be at this stage?
It will be in the liquid that's in the top, so it will come out of what's left of the tissue, just sort of the fibrey bits will stay at the bottom, and the DNA will come up and be nice and suspended in the liquid up the top.
It's early days in this research. They plan to use this test to update that data that Tom was talking about, the prevalence of clarithromycin resistance. And they also plan to test whether it will improve patient outcomes.
At the moment, we're essentially building up these methods, developing them and making sure they're going to work. And then in the future, we're hoping to start up quite a large-scale clinical trial using these methods that we've developed and using that to see if more tailored treatment would work for people with this infection. So we would test to see if their bacteria that they're infected with is resistant to the antibiotics, and if so, give them different antibiotics accordingly, and seeing if their outcomes are better than those who just get the standard everyday treatment.
And as that works, is that you're going to just get more kind of little samples of tissue that arrive into you in the lab for you to work through?
Essentially, yeah. Little bits of tissue, maybe some little stool samples.
At the moment, you're using tissue that's come directly from somebody's stomach, but the hope is that you'd also be able to extract DNA and be able to identify that antibiotic resistance in the bacteria by a stool sample, which is easier to collect.
Yes, definitely, yeah. So the top diagnostic method in the country is still taking the tissue samples, but obviously it's just not a very nice procedure to have done. They can do a test for the antigen, so a protein of the bacteria in stool. So we're hoping to be able to use these sort of DNA-based methods on those stool samples as well. So it's a lot easier to get the samples, it's a lot nicer for the patient, a lot less invasive, and hopefully a much faster turnaround time as well, so they don't have to book in for a surgery and that sort of thing as well.
And have you been able to try that yet? You know, the extracting DNA from the stool sample and running it through the tests?
Yeah, we haven't done that yet. We're planning to. We're hoping to get a few stool samples in, starting to come in the next sort of month or two from out in the community. And so we'll work through that, and hopefully that works just as well.
If the clinical trial shows that this method of testing for resistance and giving targeted antibiotics is more effective, then it could have a big impact, Tom says.
That would be practice changing. That would change how we treat Helicobacter pylori. People from New Zealand would send the stool samples or the biopsy samples to a lab that would trigger testing for clarithromycin-resistant genes, and then that clinician would get a report back saying they're clarithromycin-resistant, treat them with this, or they're not clarithromycin-resistant, you can use clarithromycin-based regimes.
And if you were to add this, molecular diagnosis step that allows you to tailor the therapy, is there a big extra added cost there for the healthcare system, or does it all even out because then you don't have a second round of antibiotics to go through for the patient?
Good question. I think that kind of analysis is really, really important, and some studies overseas have looked at that cost-benefit analysis, and generally it comes out as being less costly by doing this molecular testing for all the reasons you've just explained. First of all, you don't have to, the eradication rates are higher, so you don't have to prescribe that second line therapy. But probably more importantly is it's better for the patient. They are more likely to eradicate Helicobacter pylori, which will treat their symptoms, reduce the risk of these consequences of it. But also it's meaning they're not exposed to additional unnecessary antibiotics. And I think, we need to be saving our antibiotics for when we need them, really.
The clinical trial is currently being designed in collaboration with Dr. Stephen Innes in the University of Otago and aims to work with MÄori and Pacific health providers to ensure they recruit people from those most affected populations. Stephen and his team have also just started a pilot study checking the feasibility of incorporating Helicobacter pylori testing into the bowel cancer screening program.
So the bowel cancer screening program is being rolled out to MÄori from the age of 50 rather than 60, and so they're looking at the same time as doing a stool test for the bowel cancer screening program, they also test them for Helicobacter pylori because that's such a high-risk group. So kind of getting a 2-for-1, checking for bowel cancer but also checking for Helicobacter pylori, and then if they're positive, treating them. So we're doing two things. We're trying to improve the treatment, but also in the broader picture trying to improve how we detect and find people with Helicobacter pylori.
Ultimately, the goal is to reduce stomach cancer rates in New Zealand and improve equity within the healthcare system. And that's what drives Tom.
I think it's really, really important that we have more clinician researchers in New Zealand who are at the front line dealing with the problems that we need to solve back in the lab, back at the places like the Malaghan Institute. And I think seeing cases of things like gastric cancer or other diseases really drives that passion to go and solve the problem and improve healthcare for them.
Thanks to Dr. Tom Mules and Kate McLean of the Malaghan Institute. Now, just as changes in the Helicobacter pylori genome can affect whether antibiotics will work on it, small variations in human genes can change how people might respond to therapeutic drugs or affect their susceptibility to different diseases. One AUT researcher is keen on learning more.
Malolele, my name is Chris Boulier. I'm a lecturer at Auckland University of Technology, and I teach immunology, and I also do immunology research.
Born in Tonga, Chris's parents moved to New Zealand when he was young, to Tamaki Makaurau, where he grew up. And he didn't originally set out to research immunology.
I was particularly interested in doing research around cancer, and I found myself in a lab that looked at the immune system and how it can be used to fight cancer. And so I then discovered the immune system, and I just thought it was amazing, and I love it.
And what is it about the immune system? Because from my knowledge of the immune system, it's just incredibly complex, right?
It's very complex. I don't think we know absolutely everything about it. There's still lots to be discovered, and that's just why I enjoy it, because we need to find out more about it, and that's what I enjoy doing. Also, genetic variants. I think genetic variants is an area that hasn't really been looked at with regards to immune function and immune variation, and so that's another thing that I'm also interested in. There are good reasons to suggest that immune variants may play a role in how our immune system responds, and so that's something that I'm very keen to look at.
We all have the same set of genes written in a 4-letter DNA code. But within these genes, there are little differences. Sometimes these variations might be just one single DNA letter change. But our genes are instructions to make a protein out of amino acids. So if this change means that now the code is for a different amino acid, the protein that gets made will be slightly changed.
If that difference is in a really interesting area like an active site or a substrate binding site of a protein, that might mean difference in responses.
So these are like important parts of the protein where it can interact with something else?
Yes, absolutely. Yeah, very important parts of the protein where it could interact with another protein, or it could be an area that is a therapeutic target, for example, for a drug. And so that can then become very interesting and very important for us to know.
In particular, Chris is keen to study commonly seen variants in MÄori and Pacific populations.
One thing that we've found, and this is some work carried out by another group from Otago, is that MÄori and Pacific people have a unique genetic makeup, virtually absent in other ethnic groups. The role of these genetic variants in health of MÄori and Pacific people, what it does, we're still not sure, and that's what we want to work on, and that's what we want to find out. And so for myself, I'm particularly interested in looking at these genetic variants. How do they influence our immune system? And they make our immune system work better, or does it compromise our immune system? These are things that I want to find out.
In previous work, Chris focused in on one commonly seen variant in an immune signalling protein called JAK2. Everyone has,
the gene for JAK2, but for MÄori and Pacific people, about 20% will have a variant in this JAK2 protein, and it's in an area that is very interesting and something that motivates me to try to look at it further, is that this genetic variant is located very close to an activation loop, which is a really important part of the protein.
Now, there's a separate, previously studied mutation also in this part of JAK2, which is known to be a driver of a specific type of cancer. In this cancer, your body makes too many red blood cells, and it's called polycythemia vera.
And so we then wanted to find out, is this variant, does it contribute to polycythemia vera? And that was a lot of the work that I did throughout my PhD.
His results showed that this variant doesn't seem to be a driver of the development of this type of cancer. It's a really key signalling protein, though. JAK2 inhibitors are used to treat people with some forms of cancer and inflammatory diseases like rheumatoid arthritis, by kind of damping down the immune system.
The samples that we had in the group, participants that we had, were quite small, and so it's something that we need to investigate further. But at the moment, it's potentially protective against polycythemia vera, and if that's the case, that's fantastic. If we can figure out how it does that protective effect, maybe perhaps we can apply that to those without the variant.
So variants don't always mean bad news. Some might be helpful for avoiding certain diseases, and we could learn a lot from them. And that might be part of why they've kind of stuck around, says Chris.
Something tells me that there might be survival advantages to some of these genetic variants. And so you might imagine that a genetic variant might be advantageous against one disease, but might make you susceptible to another disease. And perhaps some of these advantages would have meant just thinking about MÄori and Pacific people sort of migrating into the Pacific. You know, maybe perhaps there were advantages around being able to travel for a really long time with little to no food, and maybe could be some of the examples of some of these advantages.
The thing is, most of the clinical trials to test the drugs we use today were done on people with European ancestry.
That's, again, some of the reason why, we don't know if these medications really work for MÄori and Pacific people, and if it does, how well does it work? And if it doesn't work, are there alternatives that we can look at? Maybe there are already other medications that they respond better to, and so maybe, we prioritise that and have those medications available here in New Zealand. And maybe perhaps there's nothing there yet that we can respond to. Maybe it could be a matter of slightly tweaking what we already have, to develop something new that can then work for the 3 million people in the Pacific.
And I know that you chat a lot with different communities in Auckland about your work, about what you do, about the genetic variants. What do you hear back from, your community and the others that you chat with? What are they interested in finding out?
Yeah, so a lot of this interest really started around the COVID-19 pandemic, as we were sort of going through different lockdowns and things like that. And I got a chance to really chat to the community about, just vaccine uptake, but also a lot of the work that I did with immune system and genetics, and they were just fascinated. They wanted to participate. How do they participate, and how do they get engaged and get involved? And so they've been really interested in biomedical research now. And so I'd like to carry out my research with the findings that I do. I like to share it with them as well. And what does that mean for you? What does that mean for your family? And what does that mean for our future generations?
Thanks to Dr. Chris Puliuvea of Auckland University of Technology. This episode was produced by me, Claire Concannon, with help from Ellen Rikers. An executive producer of Our Changing World is Liz Garten. Sound engineering was by William Saunders, and you can learn more at our webpage rnz.co.nz/ourchangingworld. You can also get in touch with us by emailing ourchangingworld@rnz.co.nz. Tēnā koe I whakarongo mai. Thanks so much for listening. Have a great week. Kia pai, te wiki.